Gracias.
Mariachi.
Days is very popular the kayak tourism here to see the sun. But at first it was a good idea for ecotourism, but now we have the same problem because it's not regulated. So
we are here in one of the Chinampas. This is Diana Laura.
hi, it's nice to meet you.
From the Autonomous University of Mexico,
Yes.
Restoration Ecology Group. And what Diana is holding is how they start to grow the plants from the mud from the bottom of the Chinampas. So they take the mud from the bottom of the Chinampas and they
Then
put it into these.
This bed, they take the mud. Esa es la herramienta.
Oh, okay.
Ese es el cuero. ¿Puedes ejemplificarlo, Adrián?
Claro.
So Adrián is taking the mud of the bottom of the canal.
Okay.
So you need to put this mud on the boat. Then you need to clean it because you have a lot of leaves and sometimes you have trash and garbage and something like that. You clean it and then you put this mud on the bed.
Okay, so that's how they collect the mud to put into these beds to grow the seeds.
They put it on this bed. We need to wait to dry the mud like 24 hours or 2 days, and then the local farmers cut the mud in very little squares that we call chapines. The chapin is the main or the principal characteristic of the Chinamperia or agriculture in Chinampas, right?
And then they let them grow in these little beds. And then when they're big enough, they plant them out into the fields that you can see back there. Okay. Refugio. There's the main canal, and so the first kind of pre-filter with plants, and the water kind of goes through here. You can see the volcanic rocks that are used to help filter the water, and then the various zones of the refuge to get the clean water. And there's a duck. The water from the canal flows into the refuge here. There's some volcanic rocks in those bags, and it goes through the refuge and is filtered by all the different plant species that they grow here.
Okay, so hi guys, we are here in the refuge. The refuge is basically second canal in between two Chinampas, or in the middle of one Chinampa. And the principal characteristic of this refuge is that we have a lot of variety of aquatic native plants of Xochimilco, and we use these plants to improve the water quality of the canal. So as you will see in your class, the principal canal in Xochimilco is very very brown, or we have a lot of contaminants in the water, and here in the refuges we have a good quality of water. And this is because of the plants, because we have biofilters here with native plants, and because we are trying to restore the trophic interactions between the phytoplankton, the zooplankton, and another species to create a good habitat for the axolot. So as you can see, we have a lot of tiny biodiversity here. We have amphipods, ostracods, fishes, little fishes.
I think I saw a dragonfly larvae in there.
Oh yes, dragonfly larva. We have another tiny animals called pulga de agua or cucarachita de agua.
So like cockroaches of the water?
Yes.
Okay.
So this is a good indicator of the water quality here, because we have biodiversity, we have life. And if we take a sample for the principal canal, maybe we don't have anything in the sample. So hi guys, we are taking here a water sample because this year at the laboratory we are doing a census of the population of the axolot. This is the first census since 10 years ago. The last one was in 2014, and we have different census in all these years. The first one was in 1998, and they found that we have 6,000 axolots per square kilometer. Then they do another census in 2010, and then the other one in 2014, and we only found 36 per square kilometer. So if you think in the first census and in 2014 census, you have a very alarming situation about the population of the axolot. And this year we are doing a new census that we start last year and we almost finish. And we have two methods to collect data. The first one is the capture of the individuals, the physical axolots in the canals, and the second one is DNA environmental analysis. So we are taking samples in the canals. We take 500 milliliters of water of the canal. Okay, so as you can see, you have the water sample, and then we put 1 milliliter of sodium acetate. And then we send these water samples to the DNA laboratory in Ciudad Universitaria, or in our principal campus. And the DNA, the principal goal is to search the DNA change of the axolot. So with this water sample, you can find a lot of species in the wetland, but our principal goal is to find axolotls DNA. So we put 1 milliliter here.
And what's the sodium acetate do?
To fijar la muestra, to conserve the water sample.
Okay, preserve, yeah.
Yeah, preserve. Sorry.
So the DNA won't get broken down by the biological processes. Okay.
And we need to use gloves because if we don't, you can find human DNA in the water sample. Yes, because I am taking with my hand.
Mm-hmm.
Yes. And so that's all. We take the samples and then we process in the lab.